Hello, I am Heth Turnquist, an investigator at the Starzl Transplant Institute, often referred to as the STI. The STI was established in 1985 in the honor of Thomas E. Starzl, a real pioneer in transplantation, surgery, as well as immunology. The faculty of STI are made up of clinician scientists as well as discovery scientists like myself, and most of us are faculty in the PMI. There are several current PMI students completing their graduate training in our laboratories, and we have a T32 in transplant immunobiology to support the development of the next generation of immunologists. Research at the STI ranges from answering very fundamental and basic questions in immunology and transplantation biology to completing translational endeavors with the goal of improving the outcomes of solid organ and even hand and face transplant recipients. We have investigators also working very hard to improve stem cell transplant for the treatment of malignancies or genetic abnormalities. There are others developing novel cell therapies for transplantation, as well as for pathologies such as autoimmunity or even tissue repair. While transplantation is often viewed as a mature field, there really is plenty of work to be done to help the more than 100,000 people each year that receive a solid organ transplant globally. These individuals or recipients will require lifelong immunosuppression that comes with it the well-known side effects such as increased infections, increased cancer, but also high blood pressure, as well as kidney damage. It really remains scandalous that the drugs we give kidney recipients what can actually cause loss of the graft they received, or can cause kidney loss requiring a transplant in someone who received a heart transplantation. Obviously, more can be done. This toxicity is a major reason that the transplantation of vascular composite allografts, such as the face and hand, are not more common. While these procedures are very feasible, they are not life-sustaining. So it is difficult to justify subjecting these individuals to these toxic drugs. Even at these toxic levels of immunosuppression, most solid organ transplant recipients develop graft fibrosis and vascular disease that will eventually cause graft loss. In the case of heart transplant, 50% of the transplants given this year will need to be replaced or be lost within 10 years due to this graft vasculopathy and fibrotic disease. There remains a significant need for innovation in stem cell transplantation as well. Graft versus host disease remains a very common lethal side effect of allogeneic bone marrow transplantation. Yet, malignancy relapse and infection due to poor graft function or immunosuppression to control GVHD remain common barriers to good long-term outcomes after this procedure. So these problems are why the transplant community really remain at the forefront of applying cutting-edge immunological tools and clinically relevant small animal models to better understand the cellular and molecular immunobiology of transplant rejection and tolerance. As someone who came to the STI as a postdoc and went from a T32 funded fellow to tenured faculty here at Pitt, I can attest to how stimulating and rewarding it has been to study the immune system as part of the STI in the Department of Immunology. So I hope I have perked your interest in transplantation as well as the STI, and that you will follow me around as I lead you on a virtual tour of the facility, as well as introduce you to a few of the faculty that may have openings in their labs for a rotation, as well as a potential place to complete your graduate degree. Now we're entering into the common space. To the left, we'll have my laboratory over here. Say hi, Galen. This is just our second bay. You can see the bays are divided up amongst primary investigators. This is David Rothstein's space. Over here we'll have Dr. Lacus' space. It is a nice way to collaborate and interact with each other, as well as share equipment resources. Then coming around this corner, you'll see we're heading into what's to us the tea room. It's a really nice space where we all kind of gather, especially during non-COVID times, have your lunch, make yourself an espresso, and just really enjoy nice stimulating views of the city. This is looking west out onto Pittsburgh. There you can see the view downtown. Along there, that's referred to as the south side and south side slopes. And then over here, it would be the Monongahela River. And then you're kind of looking down into, over here is Montefiore Hospital. And then down around us is what's referred to as Oakland. So we're going to take a right here, head into this space here, where we'll meet up with Matt Nicotra, and he'll tell us a little about how they're studying hydroctinia to establish or identify potential new pathways of allorecognition.
Hi, my name is Matt Nicotra. I'm an assistant professor here at the Starzl Transplant Institute. Welcome. You're probably wondering why we're here standing in a room full of fish tanks when we're supposed to be talking about transplantation. And so I'm going to explain to you a little bit about something that's pretty innovative that we're doing here at the Starzl Transplant Institute, which is to use an evolutionary approach to try to understand and discover new mechanisms for how transplants are recognized and rejected in the human body. So specifically, what my lab works on is an organism called Hydractinia, which you probably haven't heard of before, but it's similar to like a coral or a sea anemone in that it lives in the ocean on a surface. And as it grows, it covers its surface and eventually it bumps into other members of its species. And when it does that, it can recognize whether they're related or unrelated. And if they're related, they fuse together and become a larger animal. And if they're not related, they start fighting with each other until one of them kills the other one off. Now, if you think about it, that's sort of like what happens in a transplant, right? You get an organ from another member of your species, and your body recognizes that as not part of you and starts to reject it. So what we want to understand is whether what's happening in these animals, which do essentially natural transplantation experiments, has anything to do with what's going on in the vertebrate immune system. So these animals here, I'll take one out of the tank so you can take a look at it. We grow them on glass microscope slides. And they just live attached like this. And as they get bigger, they'll bump into each other. And under the microscope later, I can show you what that actually looks like under high zoom. And so what we're trying to figure out is how they do this. How do they recognize whether they're related or not when they don't have the kinds of the parts of the immune system we normally think about with in the human body. So in the human body, you probably have heard of HLA molecules, which differ between people, and that's what's recognized by your B cells and your T cells, and that's what triggers an immune response. So in humans, B cells and T cells use proteins on their surface that signal through different pathways, and those you can think of those as essentially things that tell the cells whether they should attack or not attack. What we found in Hydractinia is that these organisms use other proteins that on the surface don't look like the proteins that B cells and T cells use. But inside the cell, the part of that protein that sticks inside the cell has some things about it that look very similar to the parts of the B cell and T cells that tell those cells whether to be activated or inhibited, or whether to reject or not reject. And so what we're in the process of doing now is understanding how those pathways work in Hydractinia, because that could point us in new directions for thinking about how those pathways work in humans as well.
I'm stepping out of Matt's lab. You can see across the hall, this is just where our cell culture space is. This looks very much similar to any other cell culture space in the world. This is where we'll do our primary cell culture work, as well as sometimes tissue collection and cell isolations. Now we're going to step over here and go into our 2-photon facility and meet up with Martin Barscheid. And here his group is using cutting-edge 2-photon and reporter mice to complete basic discovery in transplantation.
My name is Martin Oberwanscheid. I'm an assistant professor at the Starzl Transplantation Institute, and today I would like to show you some of our research. Behind me is a 2-photon intravital microscope that is a microscope that uses pulsed laser light to excite fluorescent proteins in cells that will emit light so that we can visualize different cells in a live animal studying their cell interactions. What I would like to do now is demonstrate some of the image data on the computer screen to you. The main focus of our research is on how transplanted organs get rejected by the immune system of the body. And specifically, we're interested in how one of the major players of organ rejection, the T cell, is getting into the transplanted organ. And to answer that question, we employed intravital imaging of transplanted mouse kidneys to answer that question. What I'm showing you here is an image of a transplanted mouse kidney showing in red the blood-filled capillaries of the kidney, and in green, cells that surround those capillaries. And these cells are tissue-resident dendritic cells. These are cells that live in the kidney that are able to pick up foreign antigen and present it on their cell surface to other immune cells in the body. And the microscope here allows us to record a three-dimensional image volume that can be rendered by the software and rotated in many directions. What helps us in analyzing these image volumes is that we can create surfaces according to the signal that we're recording with a microscope. And now you can see smooth green dendritic cells surrounding these capillaries in the kidney. One of the first observations in a native kidney was that these dendritic cells are not only surrounding the capillaries, but they're also inside of those capillaries. And that is demonstrated in a different movie that I'm going to show you. So what you can see here is kind of a fly inside of the capillary, and you can see these green processes from the specialized antigen-presenting cells that surround the capillaries inside of them. This was an important observation that we followed up in kidney transplants. This movie is demonstrating a kidney that was transplanted 3 days ago, and what you can see here are blood flow and blood capillaries shown in cyan. You can see T cells of the recipient, the cells that can mediate organ rejection, in red. And you can say the specialized antigen-presenting cells, the dendritic cells of the donor kidney, in yellow. And you can see the same cells of the recipient in green. And what you can appreciate is that there aren't that many T cells around that can destroy the organ, and that there are mainly yellow cells, which are the dendritic cells that came with the kidney graft and are of the donor origin. Now, when you look on day 7 after transplantation, you can see that the picture has changed dramatically. Here again, you can see the blood capillaries and the blood flow in cyan, and you can see the dendritic cells of the recipient in green. And as you can see, they are highly outnumbering any potentially left cells in yellow of the donor, indicating that the dendritic cells of the donor left the kidney, and now we have a massive population of the transplanted organ with a green recipient dendritic cells. That was one of the first studies that demonstrated that dendritic cells of the recipient go to the transplanted kidney and then infiltrate it. And we then asked the question, how do the T cells actually get into the graft? Now, one point I would like to remark is that these dendritic cells have their origin in monocytes that circulate through the blood. And one of the other observations that we made is that these monocytes are initially infiltrating the organ, and then they differentiate into these specialized antigen-presenting cells that are shown here in green. Now, if you look at the T cells, in this video, we again have blood flow marked in cyan, and we have dendritic cells of the recipient here shown in green, and we have T cells of the recipient that are infiltrating the graft. And we made the observation that these T cells are in the graft tissue now, and they are closely interacting with these green cells. They're touching each other, and they're staying in place here. Now, if you take a closer look, sometimes you can catch T cells that are in the process of leaving the blood vessel and migrating into the kidney to cause their rejection there. And what you can see in this movie is that there is a red T cell inside of the capillary that is leaving it while interacting with that green recipient dendritic cells. And further experiments have shown that these dendritic cells of the recipient that reach inside the blood vessel are extremely important to mediate the T cells' infiltration of the organ and leaving the blood vessel and getting into the tissue. And the tissue is the place where these T cells can now mediate their effector function and destroy the graft and cause graft rejection. So overall, our research for the first time has shown that it is dendritic cells that present foreign antigen from the graft to the T cells of the recipient to mediate their infiltration of the graft and to enable them to cause graft rejection. This important step in investigating how T cells get to the graft and mediate graft rejection will hopefully enable us in the future to find ways to interrupt this process and allow the T cells to not get into the organ and cause graft rejection. And with this research, we hope in the future to be able to improve patient outcomes and improve lives after transplantation. Thank you very much.
Hey, I guess introducing people to the STI. And I know one thing from my interactions with students, people are often interested in like taking basic findings and translating them into, medical discoveries. And I just know from your work, you've had a lot of luck being a really good mentor to a lot of Department of Immunology students who have been involved into studies that have now you're translating into the clinic. And so I was just going to ask about your experience with that.
Well, we've had a very significant number of graduate students, both MSTP and mainstream graduate students in PMI, who have made very significant contributions to the development of new technologies and provided insights into the potential of immune cell therapy and organ transplantation. So in more precise terms, we've developed regulatory dendritic cell approaches to promoting improved outcomes. And by that, I mean being able to reduce and even wean patients off immunosuppressive drugs and reduce the untoward side effects that they have by administering this particular type of regulatory immune cell that we've developed from a very early stage all the way through to current clinical trials. Our grad students have been very important in working both with small animal models and also with what we call translational preclinical models. These are non-human primates that provide a more realistic model for human extrapolation. And so we have our most gifted clinical surgeons working with our trainees and our skilled technologists to translate what students have done in small animal models through research in these preclinical large animal models to validate the developments. And as we have done, then taken those developments into clinical liver and kidney transplantation. And we really are at the international forefront of these developments, whether it be gaining detailed insight into mechanisms using small animal models through real testing of feasibility and safety and efficacy in the larger clinically relevant models, to working with patients on NIH-funded research that we hope ultimately will benefit organ transplant recipients in terms of improved, safe, long-term outcomes, and eventually getting off all anti-rejection therapy, which is the ultimate goal.
Now, I know you have at least 2 or 3 clinical trials ongoing, and I know often clinical trials can be perceived as something maybe not as exciting, but I know you guys have done quite a bit of work to really incorporate some cutting edge, not only sample availability and cell therapies, but also technologies. Maybe can you tell us a little bit about that?
Well, thank you, Heath. That's very appropriate. We are able at the Starzl Transplantation Institute to apply, cutting edge technologies such as single cell RNA sequencing, image stream analysis combined with conventional, flow cytometry. We can employ techniques such as CyTOF. We can monitor T cell repertoire to ascertain whether there is deletion of specific donor reactive immune cells, and combined with cutting edge biopsy analysis, including transcriptional analysis, combined with single cell analysis, we can get a well integrated insight into how our interventional cell therapy is impacting on events that underlie transplant rejection and regulation of rejection to promote tolerance.
All right, Angus. Well, hey, it was great visiting with you. Thanks for making time.
Thank you, Heath. Always happy to talk about the work that we're doing in the STI and applying immunology in transplantation. Thanks for the opportunity.
Coming up on the east side of the Starzl Transplant Institute. You can see there's the Peterson Event Center looking east. And then again, there's your Cathedral of Learning. Over here we have Presby Hospital. And so right behind us here, we have one of the flow cytometry core facilities. Inside, we're going to talk with Dr. Camilla Booth, who will tell us about how they're using flow cytometry as well as other related technologies such as image stream in transplantation research here at the STI.
Hi, my name is Camilla Booth. I'm a physician scientist here at the STI. I came here 20 years ago for a postdoc fellowship and never left. Now I'm a junior faculty. I'm going to be showing you today this instrument. It is a flow cytometry cell sorter that has different functionalities. And again, over this last 20 years, this evolved in a very different way. What we do, I'm going to show you here, like you have your mix of cells, your whole blood, your drop-off cell of whole blood. Then you add some antibodies, and then these cells would pass through this fluidic system, and then by lasers, they're going to identify each cell what it is. If it is in a white blood cell, it is a T cell, a B cell, NK cell. And these all happen on these machines. Here we have, this instrument has 5 lasers. We can go up to 30 colors, so 30 different functionalities of these cell types. And and what we can do in terms of experiment-wise, we did in the past, we co-culture cells from a transplant recipient with its donor and put in a culture for 5 days. And after 5 days, we look at how they react to each other, how the recipient was reacting to the donor cells and peripheral blood. So then you can see each dot here represents one cell, and you can see how they proliferate, how they go against each other. So this way give us an idea how the recipient is reacting against the donor. And we can also look at functionality of the cells, not only cell types, but how functional they are. So this instrument definitely, it was a breakthrough through the whole immunology community, especially in the transplant field. What else we can do, it can sort cells. What it means, again, the same thing. You have a mixture of cells, you add antibodies, then you pass through this instrument, and according to what cell type you want, it is going to different containers. So you can put each individual cell in a different tube. So this way you can have those individual cells and look for different things. As we did it, for example, on this experiment, we have recipients were infused with donor cells for an immunotherapy. And we could track, according to the HLA of that between the recipient and donor, we could track those cells in peripheral blood. So after the cells passed through the sorter, we sorted the dendritic cells, what we call, and then we could see, for example, in yellow here are the donor cells, and in blue, the recipient cells. So then, and this is the cell type that we want to see. So then they do have different fluorochromes. That's how the lasers pick up the signal. So that's how we can divide them and know exactly what they are. And then we can see the interaction between the recipient and the donor. Then the donor is picking up, the recipient is picking up some particles of the donor, and then can give a regulation, an immune regulation, so, or the opposite, and we can try to track if what is happening is it going to reject or not, because of we could see each individual cell visualize and see the functionality of that. And this again, this comes from this instrument that has these two main capabilities. There's a sorting the cells, or just looking at what specificity of the cells. We have about 3 instruments like that and in the core that do the sorting, and we have 5 that can do the basic flow cytometry. It's very user-friendly. You do your own sample. So each researcher here, each postdoc, fellow, they can do their own experiment, and goes as far as they want in terms of science.
All right, on my way back to my office, we're going to swing in here and talk to Kanish and let him tell us a little bit about our microsurgery space. It's a really important resource, which really enables us to complete a lot of our small animal transplantation studies.
Welcome to the Microsurgery Suite. This room represents one of the pillars of the Institute. Without this room, we wouldn't be able to do our basic science research, which involves transplanting rodent hearts and kidneys. As you can see, this work is very difficult and requires the use of microscope. This specific microscope is a teaching microscope where the teacher and the student can view the same field. The skill is very difficult to master. It takes a novice anywhere from 3 to 6 months to be able to become proficient at it. That is because some of the vessels are very small, only millimeters wide, and the sutures that are used are thinner than your hair. So it's amazingly difficult to perform this task. This room also has very specialized equipment, such as the anesthesia machine to make sure that the mice are kept under for the duration of the procedure, as well as heating pads, since mice lose body heat very quickly, and as well as buckets where we can keep the mice and keep them from running away. Now, we wanted you to get an idea of what it is to transplant a mouse organ. Dr. Lily Dye here has harvested a heart organ from a mouse. You can see that the organ itself is very tiny, and just without a microscope, you will not be able to see the vessels themselves. This is essentially one of the most difficult skills that you can master here at the Transplant Institute, and it's essential to our work. The University of Pittsburgh was where some of the basic principles of microsurgery was developed by the father of microsurgery, Dr. Sun Lee, and this excellent work is continued on today by people like Dr. Day. We hope that you have enjoyed this tour and that we will see you in person very soon.
And our final stop will be this lab here, where we're going to stop and talk to Dr. Hughes, and he'll describe their work doing ex vivo organ perfusion.
My name is Dr. Christopher Hughes. I'm the surgical director for liver transplantation at UPMC. And we're studying in our lab a process called sonothrombolysis to try to increase the effectiveness of liver transplantation and prevent some of the complications after liver transplantation. One of the main problems that we have in dealing with donated livers is the fact that there may be thrombi, or what is small blood clots inside the liver that are never cleared before the liver is transplanted, and those can damage the liver later on. And so there's been a problem within the liver transplant world as to how do we get rid of those clots and how do we make it so that when we reperfuse new blood to the new liver, the liver is able to survive well and do well. And so our project that we have is to use a tiny bubble called a microbubble that's infused by the billions into the liver in a machine outside the body before the liver's transplanted. And those tiny bubbles circulate through all the veins and arteries in the liver. And then if those bubbles are submitted to an ultrasound field from outside the liver, those bubbles will explode. Billions of tiny little explosions inside the liver that basically break up any blood clot that's inside. And so we do this process over a period of a few hours before the liver's transplanted to be able to clear the liver so that when we do transplant that liver and restart blood supply to that new liver, we don't have problems with areas of the liver which aren't receiving blood because the blood clots being there. And so this is a sort of a mechanistic approach to causing tiny explosions in the liver to clear it out so that the recipient can have a nice liver which is healthy after it's transplanted. And so our project here right now, we have a rat liver that's being perfused artificially. It's alive, it's working, it's making bile and performing all of its functions. And once we have the perfusion like this going, we'll submit the microbubbles into that perfusion and then the ultrasound to clear that liver of any blood clots. And so once that procedure is completed, we'll take the liver off the machine and we'll transplant the liver into another rat to see how it does and see how it survives. And once we finish the rat studies, we'll move on to human studies and test the human liver on a much bigger scale, but with the same tiny microbubbles to see the effect there. And we're hoping that in the long run, it will improve the outcomes for liver transplant overall.
All right, go ahead and now we're going to walk you back to my office. I appreciate you coming along for this tour. Let me know if you have any questions regarding the PMI program, doing transplantation research at the Starzl Transplant Institute. I would be happy to answer any of your questions, and you can just email me at het5@pitt.edu. Thanks. Good luck with your search for graduate school.